Okay. Mattias is here. So, hello to everybody. So, my name is Marc Palardi. So, I am in charge of the webinar for EUROTOX. Can you please shut down your microphone, everybody, so we can, if it's possible. Okay. So today we have a very nice webinar, which will be given by Mattias Öberg. The name of the webinar is Navigating Chemical Health Risk Assessment. So Mattias is an associate professor at the Karolinska Institutet in Stockholm. He serves as a principal researcher and a research group leader at the Institute of Environmental Medicine in the Karolinska Institute. He's also an expert coordinator for chemical issues at the Center for Health Crisis at Department for Global Public Health. He earned a Master of Science in Biology from Stockholm University and later obtained his PhD in toxicology from the Karolinska Institute in 2003. His doctoral thesis focused on the health risk assessment on dioxin-like compounds in complex samples, which is a very actual topic. Mattias Öberg has also held various academic and research positions throughout his career, including postdoctoral appointment in WHO, European Center for Environmental Health. In addition to his academic contribution, Mattias is actively engaged in public outreach activities and recently served as expert witness in the PFAS trial that went all the way to the Supreme Court. He also holds various health risk assessment assignments, both nationally and internationally. For example, he's a member of the Nordic Expert Group for Operational Exposure Limits and as an expert in an advisory committee for setting acute exposure guideline levels in the US. He has served as a chair of psychology from 2012 to 2015, and now he's a member of the EUROTOX Executive Committee since 2018. And he's also chairing the Education Committee from EUROTOX. So thank you very much, Mattias, for accepting to give this seminar, and please shut down your microphone during the presentation. Then for the question, you can put question in the chat, or you can ask question at the end of the presentation. So the floor is yours, Mattias. Thank you.
Thank you so much, Mark, for that presentations. It's always interesting when you hear sort of your own academic history from someone else. Like, what, did I do that? Yeah, it was the years at WHO when I was working with passive smoking, and then we have moved on towards more persistent organic pollutants, and very recently the PFAS story. And I will tell you a little bit about those different items in this lecture that is supposed to be an overview of the chemical health risk assessment field, you could say. So it's not so much about my research, but more about how to a little bit navigate and understand health risk assessment. And maybe we can come back next year with webinars more directed towards smaller issues. But I'm happy also to answer questions after these 40-45 minutes of presentation. See if I can also change slides. Yes. So this is an example from a Swedish media report about 4 or 5 years ago, I think it was. It's about green tea and pesticide residues in green tea. So there was a journalist or a group of journalists sending in samples of green tea and try to analyze pesticide residues. And this was sort of the main result, that in some of the organic green tea products, they could only find very small traces of single pesticides. And they were all below the limit values. But in some of the more bigger brands, they could find up to 15 different pesticides, and quite a few of them were also above limit values. And then the journalists, of course, call around and say, okay, what does this mean? And I think as toxicologists, we all know that this is the most common question that we get, and is this dangerous? And I will comment on that a little bit later. And what's the difference between asking the question, is it safe or is it dangerous? But this was at least one example of how knowledge and risk assessment can sort of come into your daily work, even if you're not working with the agriculture or in an agency responsible for setting limit values. And how can we, what do we normally do when we get these type of questions? This was one of the chemicals. It's a post-harvest fungicide, 2-phenylphenol. And it has a defined critical effect, which is kidney failure in rats. And there is agencies developing acceptable daily intake values. And if you compare those intake values with the concentration that these journalists found in the tea bags, you could see that the concentration in the tea bag was 0.65 milligram per kilo, but there is a lot of, there's not so much weight in dried tea. So actually you need to drink more than 18,000 tea bags every day to reach this acceptable daily intake. And I've only used 2 slides, and in this slides there are several concepts that we need to understand. What do we mean with the critical effect? What is an acceptable daily intake? Is that something different from other type of values that we sometimes see? Mark, for example, he mentioned that have been working with occupational limit values. Are they comparable to acceptable daily intake values? And how do we relate those values to some sort of exposure scenario? So these are some of the things that I would like to comment on. And to give this overview, I will use the National Research Council risk assessment paradigm. It's used by the US EPA, but I would say it's a paradigm that actually holds for most organizations working with risk assessment of chemicals or health risk assessment. It starts, if we start from the left side, it starts with hazard identification, and then it a little bit divides into one aspect of looking closer into the dose response assessment, and the other one looking maybe a little bit more close into the exposure. And by combining those into what is called the risk characterization, we are able to give some sort of information to the risk management side, the blue side. And when we are moving on to that side, there is, of course, a lot of legislation. I mean, the management can't just manage risk as we do normally. I mean, take the best as we can, but they have also to fulfill different legislations. And maybe they have different options how to control or mitigate the risk. And then in the end, I will also comment a little bit on the feedback loop in this paradigm, because I think it's very, very important to understand that this is not a static exercise that you do one and for all. So you take a chemical, you do the risk assessment, and then you're ready. Because we are living in a world with new information, which changes in attitudes, so we need to be updated, and we need to have this feedback loop back to the risk assessment side. Another important and maybe also a good document for you to know about is the WHO Human Health Risk Assessment Toolkit. It's a report that was updated, so it's a second edition. I think it was updated 2021. And I would say the main difference between those two sort of documents, if you see the National Research Council as one document, this also adds on a very, very important part, and that's the problem formulation. What business are you in? What is the purpose? What is the objective of the assessment? but also what is sort of from the management side, what is accepted, what can be acceptable, what degree of uncertainty can we accept in this system, and of course, what is the identity of the chemical? That can also differ a little bit depending on how much you know when you get the question. So these are some typical aims that I have sometimes worked with. There might be other aims, but these are at least 5. With a toxicological health risk assessment, I would say a very common aim is to relate the health risk to the exposure. And the example with the tea bags is exactly that you try to get an idea about the health risk, you characterize the health risk, and you compare that with the exposure to say if the risk is high, low, acceptable, or something else. But it could also be that you want to set health-based guidance values. For example, an ADI, acceptable daily intake, is a typical health-based guidance value. It's more of a tool for management rather than the final answer. Maybe the acceptable daily intake value can be used and transferred into a limit value, a type of legislation. Is this allowed or not? But that's something different than just giving this guidance. A third aim could be investigating suspected outbreaks of illness. If you're working with acute exposure, and in these, I guess many of us live in countries where we are discussing a lot about preparedness and response and what to do with chemical releases, if they are intentional, like in terror attacks or wars, or if it's more like incidents or accidents with transport of chemicals. If you don't know the chemical, how do you start? You can sort of do the risk assessment the other way. Start, okay, what are the symptoms? And then you need to know a little bit about different chemicals, and you need to be able to compare the exposure with the symptoms and then try to make conclusions. Regulation and authorization is, of course, very important for industry, but also for agencies. Some legislations put a lot of responsibility towards the producer, while other legislations put a lot of responsibility on the governmental side. But they still need to sort of regulate and give this authorization. And at the end, also an important aspect is this internationally agreed classification and labeling, which has its own little framework and use different words so we can transport chemicals in an efficient way. If we look at the different you want to have to get the effects, but you also need to know if that is safe for the patient. So you have a lot of testing, and of course you do a lot of preclinical testing using computer models, cell models, animal testing, and then you have the different phases of the clinical testing, starting with first in human, and then maybe a small group of disease, people with the disease, and then you go on with phase 2 and 3, and then you continue with this post-market testing, sort of to follow up if there is new information. So it's a lot of data. And pesticides or plant protection products, biocides, they're also used with an intention. I mean, we are actually spreading these around, so there's a need for a lot of information. Food additives also, but maybe not always that much. It depends a little bit on how they are intended to be used. And when you go down on this list, cosmetics and hygiene products, cosmetics in Europe, for example, it's not allowed to use animal tests for cosmetic compounds. And so you have a little bit less testing. And in to the bottom of this list, the industrial and household chemicals, we have legislation that demand testing, but maybe mainly for high volume chemicals. Low volume chemicals doesn't have a high demand of testing, I would say. And in the end, we have all the environmental pollutants that we are sort of exposed to. They might be persistent or semi-persistent, which means that we are exposed. There might be a risk for the public health, but it's really unclear who is responsible. Is it, I mean, there's no classification of these chemicals that are just spread around. I mentioned classification and labeling, and I just want to show you this this list of the different classes and how we label them in the global harmonized system, the classification and labeling of chemicals. And you can see all the different sorts of toxicity. So going back to risk and risk assessment, to navigate this, it is important, although it seems very simplified, to go back to what is risk. It's a function of the consequences and the probability, and that works even if you're buying a new house, for example. You have a checklist of things that you would like to know about this house. How is the water system? Have they used the proper techniques so you don't end up in very expensive renovations? How is the economy going right now? The interest rate, maybe your personal economy, is it a high risk that you will lose your income? Maybe you need to take that into account before making a decision. And a little bit the same thing is with risk assessment of chemicals. You have a checklist, or we have a checklist, which is the target organism in this case is humans. The different organ systems, or the systemic, more complex system like reproductive toxicity or immunotoxicity is not only one organ affected, but maybe the whole system, you could say. And then the exposure, the dose, and these this make, of course, the background of the picture I showed you from the National Research Council with the risk assessment. I mean, what are the parts there? You have the hazard identification and the exposure assessment. So by combining them and including all the different questions that you could have, for example, when it comes to the hazards, what are there any target organs? Critical effect is an important concept. Dose response, susceptible groups, all these things are part of the hazard characterization. And on the exposure side, what are the sources? What are the levels? Are there any highly exposed groups, or is it more evenly spread? What are the exposure routes? It matters if it's an inhalable chemical or if you eat it or get it via drinking water, for example. And then in the middle, who is then at risk? How large is the risk, and is there any safety margin? So a lot of factors affect toxicology or toxicity. We have, of course, the chemical-physical properties like lipophilicity. I have been working a lot with lipophilic organic pollutants, quite small in size, not so polar, and not so reactive. But I've also been working with very reactive chemicals. We recently had a case in Sweden where a family was intoxicated by phosphine, a very reactive chemical. One of them died, and it took 3 months for us to understand what was the reason, because we didn't know before. Exposure, obviously very important, and also the biological system. Maybe we are affected differently in different stages of our life. Maybe there are windows of susceptibility during the fetal development, or if you have a disease that is more common in older age. Kinetics is also something that I will comment on, and sensitivity. Maybe you have some genetic polymorphism that make you a little bit more susceptible, or maybe your metabolism is not working as quick as it is for other people, so you get a higher internal dose. And there might also be a lot of interactions. This is a picture that I will show you just because I have been, as Mark said in the beginning, I was recently involved as an expert witness in a trial in Sweden related to contaminated drinking water in a small town in southern Sweden. And this was coming from a firefighting area where they have used firefighting foam containing PFAS. And this chemical then went down to the groundwater and into the drinking water. And the levels in the drinking water was like 1,000 times higher than what we normally see in Europe. And when we measured the blood of the people living in this little town, they had hundreds of times higher blood levels. So the issue in this trial was that, do they have the right to be compensated for personal injury? Because they get different sort of diseases, but it's really hard on an individual level to prove that your disease, for example, if you are getting cancer, how do you know what gave you the cancer? How do you get that type of information as an individual? I would say it's impossible to know. But maybe if you are looking through literature, you could see an association between PFAS and the disease that you have. So I will use this as an example. So when I got this question, what I did was, of course, to go through what the different methods, the toolbox that we have to check the different hazards. So there are the physical chemical properties. We know that some of these chemicals are very bioaccumulative. So the levels in the blood will just increase over time as long as the exposure continues. And we can, of course, look at the chemical structures and a little bit understand how this works. Maybe some of these PFAS chemicals can bind to specific receptors. And we know that there is one receptor in the liver that they can bind and activate, the PPAR receptor. There is also a lot of animal experiments where they have given quite high doses to different animals like mice, rats, monkeys, and follow the effects on their immune system, on their offspring. They have seen effects on the lipids, the lipids in their blood, some hormone levels, cancer. It's very clear carcinogenic compounds for rodents. So these are an important part of the information. But then there is also, of course, a lot of non-animal based models and a lot of studies in humans, epidemiology. And that has become increasingly important for the environmental pollutants. And sometimes you can also find case reports or experiments even with volunteers. It doesn't work with chemicals that are, very persistent, but it can work in low doses, for example, looking for irritation to set occupational limit values. So in those cases, experiments with volunteers can actually be quite good. And there is different types of toxicity as well. So it's not only that you have to look, you have to see for, is it a local toxicity or is it a systemic toxicity? Is it single exposure driven or more like acute toxicity or more repeated exposure? And for PFAS, it's obvious that it's a repeated exposure. They get this via drinking water every day for maybe decades. So some of the data that was collected during this trial is coming out of the population from this little town. So they have worked with register-based studies and cross-sectional studies, and both these type of EPID studies have their pros and cons. But just to take a look at what they could see in the registers, they couldn't see an increased risk of high blood pressure or intestinal inflammation, not in thyroid disease, but they could see an increased risk of a disease called PCOS, polycystic ovarian syndrome, which is a hormonally related syndrome that affects quite a few women, and the risk was almost doubled in this town. They could also see some or borderline increased risk of cancers, kidney cancer, testicular cancer. During COVID, they could follow in registers how many were hospitalized, and there were more hospitalized people in this town than in a comparable town. They could also see from some registers that more women have problems challenging with breastfeeding. There's more girls that get language disorders of different kinds. But of course, with epidemiology, it's super difficult to say anything about the causality. And even if you use cross-sectional studies where you measure things in individuals, if you measure the exposure and the effect at the same time, you can't say anything about the causality. So for example, even if you see that there are elevated levels of cholesterol, for example, in this population, you don't know if that is actually caused by the PFAS exposure. So there are definitely some good things with epidemiology. It's you could say it's more relevant since we are looking into humans, and that it is our prime target organism. On the other hand, it's very difficult to draw any conclusions about causation and common occurrence of confounding. There's a lot of factors. In this case, for example, when I gave my witness, I was questioned for like 6 hours or more than 6 hours about PFAS and the different effects. And obviously, one difficult question is that if a disease is very common, I mean, it's a public health issue normally. For example, take the increased levels of cholesterol that I just mentioned. It's very common. It means that if you have it, it's a high likelihood that you could have it for any other reason. On the other hand, if it's a very any conclusions, and that's a difficult case in a trial, I can tell you. And another problem with epidemiology is that if we have the problem formulation in the beginning, saying that the aim is to say something about safety in the future, I mean, how can you use epidemiology? Because if you see something in epidemiology, it already means that you have a problem. So you can only identify effects in retrospect. So going back to this landscape. So that's about hazard identification. But of course, the exposure is extremely important. This is the same. To the left, you have the small city, and to the right, you have a reference group, which is quite typical from Sweden. You can see in the normal population, there's a slight increase over time. So it seems to be bioaccumulative, and you see the same thing also in this town. But if you see the scale of the y-axis, you see that in the normal population, it goes between, I don't know, 5 and 20. But in the exposed population, it goes from 5 till 2,000. So it's much higher. The exposure is much higher. And that is important for risk assessment. Due to this guy, we know from centuries that all things can be poison and nothing is without poison. It's the dose that permits something not to be poisonous. This guy was trying to get a medical degree. I think he visited 5 or 6 universities in Sweden. No, it's in Europe. I mean, he had a sharp mind, but he didn't like to be part of this very hard school system of that time. He didn't really like the professors in the universities. So he had a lot of conflicts, but nevertheless, when he was in his middle age, he died before turning 50. He had already sort of stated something that has been very, very important for toxicology since. And when we work with dose response assessment, there are different ways to describe the dose response assessment. You could work with group comparisons. You have a control group and you have exposed group, and you try to see if there's a significance. Or you can work with a more mathematical modeling approach, the benchmark dose approach, which will give you maybe a better overview of the variability in your data. And it's also more comparable because you can decide what response level you think are important instead of just taking the response level that happens to be in the experiment. And from dose response, to understand this, you also need to understand a little bit about toxicokinetics. I like this pictures. It's from Arizona. It's the Biosphere 2 project. Maybe some of you have heard about this. It's a quite fascinating project. So 8 people, 4 women and 4 men, went in to this greenhouse that was specially built to be isolated for 2 years. And they should sort of only grow their food themselves, not using any pesticides. So it should be really, really organic and nice. And it's a way to sort of see if we can take humans maybe to another planet or so. So it's that type of experiment. And they measured also then the lipophilic pollutants, because all these people going in, they had levels of PCBs and DDT, dioxins, those type of pollutants in their blood. And then after the experiment, they measured again to see what happened if you live for 2 years in this super clean environment. And the result was quite surprising. Actually, the levels almost doubled in their blood. And nobody first understood how could this happen. I mean, they were not allowed to use these chemicals. They have really been working hard to get the food. It's hard to grow your own food. So what happened was that they actually didn't get enough food. And this is a picture of the medical doctor. You can see him on this picture. You see him in the middle of the upper row. And here is the sort of before and after picture. And as you can notice, there have been 2 years of food shortage. And the first energy reserve that the body uses is, of course, the body fat. And the body fat is where all these lipophilic chemicals are diluted. That's where they sort of stay. So when you take away the body fat, they have to go somewhere else. They can't leave the system because they are very bioaccumulative. So they have a very long half-life, much longer than 2 years. So they have to go somewhere else, which means they go to, for example, the blood, meaning that the concentration increased, although the amount was more or less the same. I think it's just an interesting case about showing the importance of knowing toxicokinetics, absorption, distribution, metabolism, and elimination. So these are important questions both to understand the toxicity, but also to understand the exposure. So that's a link in between. And just give you some examples then of the chemicals that they studied in this Biosphere 2 project. This is an old study from 1981, where they tested different PCBs, polychlorinated biphenyls. And there are 209 different congeners, and they gave this to rats and just saw, okay, in the food, and see how much can they absorb. How efficient is the absorption of these chemicals? And you could see a very, very clear trend. When they get more heavy, when they get bigger, the absorption efficiency goes down. So that's one example of the importance for absorption. Okay, how are they distributed? As I told you with this picture, lipophilic chemicals are mainly stored in lipophilic components of the body, meaning that fat, parts of the skin, and of course maybe a little bit more lipids are in the liver is in comparison to blood, for example. And you can see this is an example from a study with monkeys, given also a PCB congener. And in the beginning, you see high levels in the muscles and blood, and then after a few days, it goes all into the fat and the skin, and very, very low levels in muscles and blood. And this can be modeled with pharmacokinetic modeling, and that can be a tool for risk assessment, trying to translate sort of what you know about the exposure and what you know about the effect. What you know from the intake, maybe you can foresee how much will actually end up in the liver. And if the liver is an important organ for the toxicity, it is important to know how much will go there. Another thing relating to kinetic is the half-life. This is TCDD, the most famous dioxin chemical, and the apparent half-life, meaning the half-life that you measure in human blood. What happens is it's not breaking down the TCD. It's just that it sort of goes out through the feces a little bit every day if you don't continue the exposure. And this was measured after the Seveso incident in northern Italy, a terrible chemical disaster in the mid-70s. And then they could follow the people living there for many, many years and see how their blood concentration went down. And they could then use this to measure the half-life. So sometimes chemical incidents can be very informative. So it's important if something goes wrong to learn from this, because that information can then inform other parts of toxicology. Typical questions related to toxicokinetics is the route of administration, how much reaches the systemic circulation, how is the compound distributed in the body, for how long time does the compound persist in the body, the half-life, are there any important metabolites? I mean, some chemicals are not that stable as the PCBs or dioxins. Maybe they are reactive. How can they be eliminated? Does it accumulate? And these questions can also be important if you want to understand and help if someone gets intoxicated. And that happened with the chemical that I was talking to, the TCDD. This is Viktor Yushchenko from Ukraine, already back in 2004. I mean, it has been a lot of conflicts between Russia and Ukraine for decades. Maybe we have only seen the worst part now the last 2 years, but as you know, the war has gone on for 10 years, and even 10 years before that, it was a lot of conflicts. So the Ukraine politician was poisoned by TCDD during the presidential election campaign in 2004, and he got this very typical chloracne, very high levels of TCDD. And I have discussed also with some of the researchers involved in trying to answer this question: how can we get rid of persistent and bioaccumulative compounds. It's not so easy. I mean, how do you could you make some sort of liposuction, or they're really trying different ways and not just treating the symptoms. So elimination is important. Okay, how far have we come now? Hazard identification, yes. Dose response, exposure assessment, okay, risk characterization. Then we come back to this word that I mentioned in the very beginning, a critical effect. The first adverse effect, or maybe it's known precursor, that occurs as a chemical dose increases. And this is, again, example from TCDD. So if you start with sort of low adversity, at low concentrations, you could see enzyme activity, vitamin A levels changing. And then if you get higher up in the dose, you could see reprodevelopmental effects, immunosuppression, cancer coming. If you increase it even further, you could actually push it all the way up to lethality. But when you're doing risk assessment, you're trying to find what is the first adverse effect. Is enzyme activity adverse enough? Or maybe vitamin A should be considered adverse. It's a very important vitamin, almost as a hormone. It has its own receptors and so on. Or should you work with the reprodevelopmental effects that you can really see in a intact organism? So that's important to define the critical effect. And when you have defined your critical effects, you have to look for a critical study, maybe as a group of studies, to identify sort of at what dose do you see this critical effect? Where does it start? And we call that the point of departure, the dose response point that marks the beginning of this sort of low dose extrapolation. And you can use different ways to define this point of departure. It could be the no adverse effect level, or it could be the benchmark dose. And there are different ways to express this point of departure. The next step is to deal with the uncertainty. And we use assessment factors, or it's very commonly used. You can use other techniques as well, but this is the most common one. Trying to deal with the uncertainty using assessment factors. And they can be divided in different ways. It's important to know that they can be related to the intraspecies variation. For example, if you have an animal experiment, it's of course, or if you have different humans, the intraspecies variation between us, there's a lot of variability. We need to take that into account. And if we have animal experiments as our if the point of departure comes from animal experiments, it's also important to understand the interspecies variation. These factors have also a lot of different names in different legislations. It can be called assessment factors, safety factors, uncertainty factors, modifying factors, adjust. Okay, I have a few more slides, and what we are trying to do in risk assessment is often to derive some sort of guidance value, like the tolerable daily intake, or the acceptable daily intake, or the derived no effect level. It has also different names. This is the definition from WHO. It's important to know that if you're talking about TDA, it's a lifetime exposure. But how do you characterize the risk? It means that you need to compare the identified hazards with the exposure. It can be set by a margin of exposure, like the tea bags. You could say that as an example. You compare the TDI, or the acceptable daily intake value, with the exposure, actual exposure or model exposure, to get an idea. Sometimes we work with the whole populations. You could see that a certain percent of the population is above this value. And then the next question for the risk manager is, of course, is this acceptable? Is 5% or 10% of the population acceptable to have in this sort of risk situation or not? Where do we draw the line? And the risk managers, they have also different options. They can ban the chemical maybe, or maybe they can set a limit value or a guidance value if it's more soft working. They can also give information. Say, okay, we should try to avoid this. Or dietary advice. Avoid this fish from this lake because it's polluted. Or don't eat it more often than once a month. It could also be that they have their own sort of risk control plan. So you have some sort of system, maybe a producer of some food item, and they have a self-control system and they need some supervision. So there are different ways to manage risk. I would say the main two differences that I see as a toxicologist, if I want to be sort of seeing into the future, or if there is something already happening, if I want to be protective, or if I just want to be predictive. The important thing is then the difference between if the first to be protective, like, is this safe? That's the main question. In the other situation, it's more, is this dangerous? How dangerous is this exposure? And we need to balance those things. If we're working with, for example, acute exposure, a release of a toxic cloud of something, if we underestimate the health risk, it can result in severe health problems, of course. But if we overestimate the health risk, it can result in big management difficulties. Maybe we need to evacuate the whole city and people would die just from all the traffic jams, or it can be other type of problems. And that's a decision for the risk management to be aware of. And we have some current challenges in the risk assessment. And this is probably something for coming webinars in this EUROTOX series. Late effects of early exposure, endocrine disruptive effects, uncertainty, individual variability and sensitivity, the whole mixture thing, the cocktail effects, the demand for animal-free methods, and the need to speed up the process because we have so many chemicals and so little information. So all these are challenges that is not really taking care of, I would say, in a good way at the moment. My last slides relate to my experience in the PFAS case. And it's why did it take so long time for our system or systems to find out about this problem? Can we learn anything? Can we have anything in the feedback loop? And moving to this slide, I think that our regulatory system sometimes are producing ignorance, and we need to think about this. We tend to think that existing chemicals are safe because they are already existing. They're already out there, so they can't be problematic. We think that low volume chemicals are safe, but actually the low volume chemicals is just a group that we give low priority because we think it's better to start with the high volume chemicals. It doesn't mean that they are safe. And they can also be very important for the mixture concept. We work, as you have noticed, one chemical at a time. And we want the polluter to pay for the risk assessment, meaning that we give them a lot of responsibility about their own products, and they have a large incentive to not finding effects. So that can be a problem. And there are more problems like this in the regulatory structure that we can try to learn from and to fix. But also in the academic structure, which I belong to, we have this publish or perish system, meaning that maybe we need to publish things quicker than we can really think them through. Sometimes maybe over stressing health risks, because it's easier to publish a sort of an effect than publishing a study that doesn't find anything. So we also have our work to do in the academic structure, because we are also sometimes sort of producing ignorance rather than knowledge. So with that, go back to the first slide, and I'm happy to answer any questions or ideas for future webinars. No, I can't hear you, Mark. I think you're muted.
Thank you, Mattias, for the very nice presentation, very complete overview. And I'm starting with a question. I have two questions in the chat. First one with Rafat Fares from ERBC. Quite a provocative question from Rafat. Thanks for the talk. Given the limitation of epidemiology studies, do you believe it is worthwhile to conduct them?
Ooh, that's a tough question. I mean, I think we need to take advantage of the new statistical methods that come around, but maybe we need also to plan them in a better way. When we have an incident, for example, it has been very valuable to follow up, to learn from this, both to see the critical numbers like half-lives, but also to understand what type of effects that we can't detect in our lab systems. For example, learning difficulties, or these type of how to say it, you couldn't mention, maybe they are subtle, but they can influence the human health a lot. If you think about health, then more than just a disease, or the function as a human, we need to bring the humans into the risk assessment. So I think it's worthwhile, but sometimes we shouldn't say that. I mean, a problem is that they are so weak when it comes to causality. So if we only have human data and say, okay, if we can't see anything in humans, then there is no risk, I think we are making a big mistake. We should definitely use the experimental models. And now we also have a lot of new approach methods that are more human relevant. And I have a big hope that those will actually complement and bridge the gap between animals and humans.
Thank you, Mattias. So second question is from Anne-Sophie Morisseau. Thank you for the speak. For the computers models in silico assessment, do we have example of tools that are relevant? We are using Viga, ToxT, ToxTree, Quasar Toolbox, and LeadScope, but this is sometimes difficult to know if all toxicological endpoints could be covered by this tool. She said that I know that mutagenicity can be well studied thanks to this tool, but it would be interesting to have your advice for all the other toxicological endpoints.
I will not be able to give a really good answer because I haven't worked with so many of these models myself, but I of course can comment on that, the fact that for a model to work, you need to have a good set of data to train it, and that's much easier for mutagenicity because it's easy to test in vitro, and it's also easy to understand sort of what are what type of chemical energies needed to create a mutation or a break of the DNA molecule. It's much more difficult when it comes to complex things like the endocrine system. I mean, you can quite well foresee how it will bind to a receptor, but very, very small differences in the chemical structure can make a huge difference in potency. So my experience is that it works quite well for screening, to at least get if you get a hit, maybe you can sort of look a little bit extra for something, but to only trust the models, I think they need to be combined with other type of, for example, physicochemical models that are linking also the toxicokinetic behavior. It's not a very good answer, but it's at least a little bit.
Okay, so a question from Zoe Carlo. So big thank for the presentation. Do you have examples of late effects of early exposure and how late it is?
I mean, one example could be this defects that we discussed a lot in the PFAS case that I mentioned in southern Sweden. You see an increase of PCOS, and this is an effect to difficulties being pregnant, which means that you will not notice until you're 30. But the hormone system can be affected already when you are, in the fetal stage and growing up. You can sometimes see this as girls having, a little bit more body hair than normal, which can be, of course, can give a lot of comments in school. We have seen examples in Sweden when children are bullied for this type of things. But the effect itself, what can be sort of when maybe you go to the doctor, say, why can I have difficulties being pregnant? It can only be seen when you are, trying to get pregnant decades later. So that's one example. Cancer is, of course, another one. It takes decades sometimes to develop. Even if you, of course, have different hallmarks that you need to sort of click to get the cancer in the end, it's a long lag time.
So we have two other examples for you. One is benfluorex, which is in pharma. It's a compound that was used in diabetes, and it was used also to reduce weight. And finally, at the end, about several years after, people had some pulmonary hypertension. And so it is responsible probably for 500 to maybe 1,000 deaths due to benfluorex exposure. That was still a big issue in France, actually. And the second one is DLT-cybestrol, that we use in pregnant women to avoid abortion. And so after the babies were exposed in utero, and they had some vaginal cancer, but also some issues with reproduction and development of the reproductive function.
Yeah, so I think that the endocrine system is actually something that we know. It's used to develop the body, and all these changes are driven by very strong sort of hormone receptor feedback loops. And if they are affected, it can actually change the sort of the road forward.
So me, I have one question for you. So maybe someone have some other question. Do you have other question on not in the chat, but you want to ask your question directly to Mattias? Me, I have one question for you, Mattias. It's always the link between the hazard and the risk, and you really presented it very clearly. And of course, the importance of toxicokinetic and exposure. But me, I have a question about what you said. What is the marker of the early marker of disease? When you said, okay, cytochrome P450 induction or vitamin A level modification. So it's always the same. Do we, in epidemiological studies, we would like to have the nice
it's always very important to define your sort of adversity. Where do you put? And it's important when we do risk assessment to express that clearly, instead of just sort of saying, okay, this was the first effects, and then it's like, okay, but we had other effects, but we didn't consider them. Because if, for example, the vitamin A effects, we are working now with a study where we're trying to develop relative potency values for different PCBs in relation to vitamin A changes. And we argue in that paper that the vitamin A, we know from other areas that this is a very important type of a group of chemicals, like hormones, that can affect, for example, fetal development. So if we are moving this very important chemical around, that is sort of an adverse effect in itself, because the body tries to control this very, very hard. The same goes with many of the endocrine disrupting chemicals. On the other hand, you can of course argue that a lot of things are changing in the body as an adaptive response to the environment. So it's really a living discussion, but and I'm not sure. I mean, we shouldn't wait until we see a clear disease, because we are then so far off into we are almost at the hospital when we can do that. And we can't wait until people are dying from any reason and then sort of try to go back and understand their whole life story. We want to have safe chemicals for the future. So
yeah, I fully agree. It's only a question to agree on what kind of biomarker and what kind of key characteristic of the compound, because I know also the concept of key characteristic is interesting, because for example for PFR, okay, it is clear that immunosuppression is a key characteristic of a carcinogenic compound.
And when you see discrepancies between different expert groups, it's quite often that they don't agree on this. And that's also why I think it's important to have multiple expert groups. Sometimes it's annoying because they come to different conclusions, but I think it's actually there where you can, if there's a big discrepancy, it means that there is an important question that we haven't solved. It's a call for research.
Yeah, that's important. So in the audience, anybody has any more question for Mattias? Because it's already 10 after 1. So any question from the audience? No, I've got in the chat some nice comments. Thank you very much. So that are saying that it's a very nice presentation. So if there is no more question, I fully agree with all the comments. It's a very nice presentation, Mattias, very interesting, very instructive, and right in line with the webinar from EUROTOX, the spirit of the webinar from EUROTOX. So thank you again, and I hope you enjoyed the presentation, and I wish you a very, very nice day. Okay.
The same. Looking forward to see some of you in EUROTOX in Copenhagen.
Yeah.
Bye.
Bye-bye.